Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Lili Wang, Ph.D. She's an instructor at Mount Sinai in medicine and infectious diseases. So, Lili, thanks for coming. How you doing?
I'm good. How are you?
Yeah. Tell me a bit about your background. You're an instructor. You have a Ph.D. What's your background in what you teach, and how long have you been at it?
I have been in the virology field for about 14 years. I was doing herpes virus in my Ph.D. program, and afterwards I came to New York at Mount Sinai School of Medicine doing HIV cell-to-cell spread. And at present, I'm still working on HIV in Dr. Benjamin Chen's lab. And I also mentor some PhD students for their lab training and techniques. And that's my work as an instructor. About this.
Okay, well, very good. Are you doing any research, or is it mostly teaching? What are your day-to-day work?
I did 80% work in research.
Okay, yeah, and you got to do a little teaching on the side. I got you. Excellent. So what is your research about? What are you trying to figure out about, I believe you said HIV?
Yeah, people usually think HIV transmits in the form of free viral particles, but not so many people know that HIV can also transmit in the form of cell-to-cell, that it's through a very fantastic structure called virological synapse. And if you have a microscope and with a fluorescent, you can see there is a beautiful button structure formed between the infected cell and the target cell. And you can see the virus material transfers through the button into the target cell. That's very amazing. And maybe in the future, people should include this form of transmission, so for the vaccine design and the drug, and that might be more effective.
So how specifically does the mechanism work? Would you mind talking about it for a moment?
The HIV has only one surface protein that we know called envelope protein. And this envelope protein can interact with a target cell surface receptor called CD4. So the CD4 envelope interaction will trigger the formation of virological synapse. And in this way, the virus can more specifically and more efficiently targeted to the uninfected cell. And at the same time, without detected by the immune system, or less detected by the immune system. So that's a way that's highly efficient and very smart.
Oh, wow. Well, if you wouldn't mind, I'd like to have you back in the near future to talk about HIV specifically and this research, if that's okay with you.
I think so.
Okay. Yeah. No, that's fascinating. Just one more question about this synapse effect. If you look at it under electron microscope, does the virus envelope fuse with the cell that it's attempting to, inject its genetic material into, or how does it act as a synapse? Why do they call it that?
Because it really looks like adhesive structure. And if you have the fluorescent proteins enlighten the envelope protein or the gag protein, you can see a very specific structure that connects the donor and the target cell.
Okay, gotcha. Well, if you're ready, let's get on to the questions here. First one is, do you believe that viruses are alive? And why or why not?
It's hard to say because the virus cannot live without a cell, but it can replicate. It can make thousands and millions of copies of itself. In this way, it's like a live thing. But without a cell, it cannot do anything. And it feels dead in this way. So it's hard to say.
Okay, I just wonder, if I've given this example to a lot of people. If I look at a seed, and then I look at the plant that comes from the seed, the plant is obviously alive, but is the seed alive? Is it contingently alive when it's in the right soil with moisture and everything? So when we think about viruses, if we think about the virion stage, people say, oh yeah, it's not alive. But once it enters a cell, or when it fuses and enters, now all of a sudden it seems to have all the hallmarks of being alive. So perhaps they're contingently alive. What do you think?
Because the virus take advantage of the cell mechanism to replicate itself, and it has all the elements for its early and late expression of a series of proteins. That's a cascade. So once it enters the cell, the whole process begins, and it's very delightfully controlled, and in a time and spatial very coordinated manner. And that's I think that's thousands of years of evolution made this to realize this whole thing.
Viruses have many different entry mechanisms. Some are endocytosed. Some like a phage, like a T4 phage, will have tail fibers that will touch the membrane and screw itself in. And others I've seen they'll like bite onto the pilus of a bacteria, and when the pilus is retracted, it pulls the virus in. How do you think, and why are there so many different mechanisms of entry that are so sophisticated? Where does that come from?
But there are different forms of entry, but the essence of it is you have to have a receptor. Usually the envelope, every virus has its specific receptor. That's make the that make the species have different viruses. Like a human virus may not infect a cat like this, and there is limitation of the species because the specificity of the receptor on cell or on any host surface.
But how do you think that all these different mechanisms evolved? I mean, do you think it was just blind random mutation, or it's deliberate?
It's definitely not deliberate, and I think I believe everything in virus is very carefully selected, because that's behind that's many years, thousands of years or millions of years evolution. So nothing in virus in such a delicate structure with all the mechanism in it cannot be just that randomly. It's all selected in one direction for its aim to suitable for the propagation of the virus.
Well, if like some viruses will get inside a cell and multiply and blow it open and kill it, they'll be lytic. Some are either lysogenic or latent, so hang out in a cell and maybe even be beneficial to it. I don't know, for years or even the lifetime of the organism. And then sometimes they'll change their behavior back to lytic and they'll blow it open. And then some, like retroviruses, can endogenize into the host's genetic material. So how and why are there these different mechanisms of, I guess I'll call it viral life? Like, why does these three different things happen, and what governs whether one thing's happening or not?
There are many viruses have a latency period. Like what I did previously, the herpes virus can also have a latency period with episome structure in the nucleus. And HIV also can latently infect the host cell. And once there is a trigger, and the virus can again enter the lytic cycle and produce thousands of viruses. And I think that depends on the environment of the virus. If the environment is good, is good for its proliferation, and there is a trigger, it can detect that signal and tell the virus that it's a good time for you to replicate and produce thousands of your copies. And then the virus got that signal and get triggered, and then enter the lytic cycle, then kill the host cell and produce thousands of them. That the latent virus is waiting for a chance. So their ultimate aim is still replicate more, get more copies, get the genome, the information of the virus itself more.
Yeah, but so what do you think is the sensing apparatus of a virion versus the sensing apparatus of just the naked RNA or DNA, the genetic payload of the virus? How can just a string of RNA or DNA have a sensing apparatus where it can do all these complicated things? It's weird, you know?
There is a mechanism in that. So in my previous study in the herpes virus, there is a molecular switch. It's works like a switch. It's called a transactivator element. When there is some proper signal outside, like a heat, it's very hot, the temperature is hot, or there is some change in the iron concentration, something like that, and it will activate the transactivation element. And this element, just like a switch, will turn the signal on, and the virus will begin to transcript its genome and produce more viral proteins and enter the cycle. So there is, I think many of the viruses will have a switch similar to this that can sensing the outside signals.
Do you think viruses have a sensing apparatus when they're the virion stage? You know, its capsid,
Yes.
or tail fibers, things like that, if it has any?
It may not be in the protein form. Sometimes it's in the, it's like in the nucleotide form, like a transcription. You need some you have promoters there, right? And some host proteins, or usually it's host proteins, and can bind to that part of signal and then activate the transcription. That's all by chance, by the change of the outside.
Yeah, well, okay. If I had a cell and I sucked out all the contents so that it was pretty much empty, and I just had the membrane intact with all the receptors and everything, and then if I introduced it to a virus, do you think the virus would fuse and enter, or do you think it would sense something's wrong and abort and not enter and not complete its injection of genetic material?
That depends on the virus entry mechanism. Usually the virus entry by binding to its cell surface receptor and then fuse by its own, like HIV, it needs a CD4 and a co-receptor, and then it can fuse. But even like this, it can enter the cell, can fuse with the cell and release its nucleus into the cytoplasm. As you said, there's no cellular components in it already. This will not lead to a productive infection. The virus will die. No, it's not called dies, will not replicate.
Okay, so you think if a molecular component is missing, that would make it stop, but it's not through any sensing and discernment and then change of action.
Yes, because the virus needs a cell mechanism to replicate.
Gotcha. Okay. Let me see. What's I'm sure you've heard of quasi-species. When we're infected by a virus, how much diversity is there in the genetic code of the virus that's infecting us? And do you think that a virus somehow has like an identity? I know I'm this particular sequence, and these other quasi-species are close enough to me that we'll all infect or will kind of fight each other. Have you seen any examples of viruses, once they infect a cell, there's a competitive type mechanism where they stop another virus from infecting the cell?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Another different virus, or another virus that's just another same virus.
Well, once a cell is infected with HIV, Can it prevent superinfection? Once a cell is infected by herpes, can it prevent other infections?
Yes, in my research, the HIV, for example, when HIV enters a cell, it can downregulate the surface CD4 expression. That means if another HIV virus want to enter the cell, that would become more difficult because the surface receptor is downregulated. There are not so many CD4 there.
So why do you think that would happen? I mean, it's like a dog guarding a bone and going, stay away. How would a virus do, how and why would a virus do that?
Because usually if one virus can successfully enter a cell, it can start its productive infection already. It's not necessary to get more copies of it enter one cell. You can save the virus to another cell. So this mechanism ensures that the virus can infect as much cell as possible in this way.
Okay. 1 second, let me grab another question. Do you see that other processes in cells and bacteria that are virus-like? Like if you look at a plasmid, they can be given off by a bacteria, they contain genetic material, they can enter into other bacteria, they can change the bacteria's gene expression, etc. They can even, change its DNA. Same thing with extracellular vesicles in people and other creatures. Do you see these as viral-like mechanisms, or are they, completely independent?
Yeah, the plasmids, yes, from the bacteria, yes. That's a very amazing kind of, genomics information that can replicate, but it's not a virus. In my experiment, the plasmid usually is manipulated by transfection and to enter the cell. And in the nature, I'm really not too sure how the plasmid transmit through one bacteria to another bacteria. It should be a different mechanism from the viruses. And the plasmid seems a shared information between the population of the bacteria. It's a beneficial one, I feel.
Yeah, the reason why I say this is that, viruses are tools. They're used by bacteria. They'll take pieces of their DNA or RNA and incorporate it for immunity. I've seen examples of bacteria that take viral DNA and recreate the spike protein on their own membranes and use it to poke holes in their enemies. So I wonder if all cellular life, bacteria, our cells, viruses, etc., if all cellular life can use viruses as tools, and viruses certainly seem to use cellular machinery to its own ends, as its own tool. So that's why I wonder, if you look at, again, extracellular vesicles, they're virus-like. They may not be exact viruses in themselves, but I wonder if this is just another manifestation of this trading of ability and this use of tools.
Yeah, that sounds amazing. I'm not so familiar with that, but you said this information is very interesting. I think sometimes the information of the genetics might be both beneficial for both the host and the viruses or the plasmid, as you said. And it's like, the CRISPR mechanism, the bacteria can take in the virus fragments and store it in its own genomes. And the next time the virus enter, there will be a defensive mechanism against that virus. And But
well, if you look at all the phages, let's say, that prey on a given bacteria, we call it their phagome. I'll just make up that word. It doesn't exist. Do you think that the phagome of a bacteria, contributes to its immunity and is like an integral part of its immunity? And what other roles might it play for the bacteria?
Yeah, I think you're right, because as a common knowledge that we know that if you want to find a phage against one bacteria, you just look for it around the environment where this specific bacteria locates. They are always together, so they can coexist for such a long time, and there should be some mutual beneficial effect there, right? There's a balance between the bacteria and its phage, and both of them can exist and then co-evolution, right?
Yeah, like cholera, when it's infected by a certain virus, now it has the ability to be pathogenic to people. So I just wonder if our virome, we can't see it, I guess, but I wonder if it could confer beneficial effects to our cells, because a bacteria's phagome seems to do that, whether it's the bacteria plucking elements of the viruses that it wants as tools, or, the virus is influencing the bacteria. I wonder with our own cells, our somatic cells, if our virome plays a similar role.
I think so. Our ancestors, when they are infected by a certain ancient virus, sometimes they can store this information into the genome. In fact, inside the human genome, a large amount of the sequences from the endogenous retroviruses. There are many of them, maybe can come to 10 to 15% of the whole genome. They are in fact from the ancient retroviruses. So I guess they might, maybe not only retrovirus, maybe there are also other viruses. I think that the storage of this information may help the human beings to fight against those species of viruses better with an unknown mechanism, right?
Okay. If I think of a virus infecting a cell, I think of like one virus and one cell. It infects it and that's it. Do you think that there are cases where multiple viruses will, let's say, attack a cell at once and they'll coordinate entry? Are there any cases of where 2 viruses are needed, or 3 or 100 are needed to get into a certain cell to infect it? And do they coordinate action?
I'm not sure if that exists in the physiological conditions, but in the lab conditions, sometimes we use very high titers of virus to infect a cell. Usually it's a saturated concentrations of virus and infect, and there should be. In fact, we did both green. We marked the virus with green protein and the red protein, and you can see both of the viruses enter the same cell because you can see both color and coordinate. As I said previously, we know the one virus enter, like HIV, it entered and it can downregulate certain surface protein of the infected cell. I think those proteins could be beneficial or detrimental for the other virus to enter. So there is a mechanism of some. The entry of virus and the expression of certain viral proteins definitely will remold the environment inside the cell. Yeah, so it might be beneficial for another or not. That's a complicated thing.
It is, yeah. I know. All these questions are. I know they're they're really tough questions, but why not? So okay, just a few more. So do you think that? What do you think is the interaction? Let's say in our guts, we've got our own somatic cells. We got our virome. We've got our microbiome. We've got our mycobiome. We've got phages all over the place. What do you think this interaction is like in our guts, let's say, where a lot of the activity is happening? Are our cells communicating directly with phages or our virome or our bacteria? Do you think like all these parties are communicating and either working with each other or not, or fighting each other? Like what do you think it's like in there? if we miniaturized you and put you in the gut, like what would you see with all these players hanging out in there?
I think it's already established information about the gut with bacteria. There's the bacteria. There are different species of them. There are several of them is beneficial for the human being, and it can be deemed as part of the human body, in fact, because it can, like the E. coli, the balance of the bacteria in the gut. If it's disrupted, then people will get ill, get the the diarrhea, something like that. And I believe the virus might also play a role in that, but it's very well balanced or adjusted. It's just like the bacteria. There should be a balance there, and you cannot disrupt it. If it's disrupted, the balance, and it might cause disease. But
Okay.
I really don't have an example as my prediction from the bacteria.
Yeah, I mean, it's hard to know. I just wonder, like, if you think about it, what do you think is going on, Yeah, I just wondered, again, is the communication amongst, all the players, or is the communication only amongst some of the players, That's why I wondered, so. Yeah, I guess I don't know if anyone knows at this point. It's so complicated in there. So, okay. What do you think came first, viruses or cellular forms, and why, back in evolutionary history?
I still feel cellular life forms begins first.
Okay, but why do you think? Why do you think viruses couldn't be first?
Because viruses need the cell mechanism to replicate. So it should be the cellular life forms comes first. And the virus might originate from some kind of, you mentioned, kind of a plasmid. There's information transmitted between the cellular life, like the original bacteria, something like that. And later, these plasmids, like things like material, then they gain the ability to assemble, to produce the structural proteins and assemble themselves into a virus-like thing. So that's a later thing, and make them easier to transmit between the cells. So I think the very original, the viruses might be just a simple plasmid.
Oh, do you think viruses evolved from a plasmid that went
something
crazy
like
or
that.
adapted?
Yes.
Interesting. Okay.
It's not confirmed. That's only my assumption.
Oh, I know. Yeah. This is all speculation, but that's okay, Yeah, I try to do questions that no one knows and no one may figure out for many years, but I think the speculation is really informative. It gives, readers and listeners ideas on experiments they can do.
So these questions are good for us, the young scientists, I think. Usually the famous professors, they are very, very alert to these things. They will not come to a conclusion unless they saw the data. But for the people like us, the young one, we like to imagine and see what we're curious about everything and seeing what it looks like, at least in imagination.
Yeah, that's good. That's good. So just a couple more questions. Do you think, what role do you think phages play in antimicrobial resistance, in bacterial resistance to antibiotics? What role do phages play, like the phagome of a bacteria?
Just as you said, and as we mentioned before, the virus infection to a bacteria might lead to the bacteria store the information of this specific virus and then render it new features against this kind of virus. And it may also get the information from the virus that might be, introduce other features like the antibiotics ability. It's possible, quite possible.
Yeah, I wonder if you perturb a bacterial population with an antibiotic, if that, again, some will die, some will not, but how does it change the phagome? Do phages suddenly come into play and, the bacteria says, I need a defense, and it looks in its phage library, quickly turning the pages and grabs, some genetic material from one of the phages and incorporates it, and now it can fight off the antibiotic,
Yeah, that's quite, I think this presumption is amazing. Yes, that's quite possible. Like if you have the pressure, like you add antibiotics to the bacteria, and most of them died, and only those with resistance lived. And I think those one has a plasmid with antibiotics resistance. And I think the replication of these bacteria, the left bacteria, also have the plasmids propagated, has more of the plasmid, and we can transfer this information to the neighbor bacteria, and yes, get the whole population immune to the new antibiotics.
Yeah, maybe, yeah. Okay. Well, I guess last question. Overall, what roles do you see viruses playing in evolution, adaptation, speciation?
Yes, that's a great question. So usually people deem the virus as a bad thing, as cause diseases, cause many troubles like COVID-19. But in fact, if you look in the evolutionary view, I think the virus plays a very important role, and it contributes to it a lot. We have to thank the viruses. They molded what we are right now. We are now resistant to many different infection, either bacterial or the viral infection. I think that's we owe a lot to our ancestors fighting against all these viruses. Yeah. So I think because of this competition between the host and the virus, that results in the modern world of the whole biosphere.
Okay.
Yeah, there is the virus has to, you have to say it's a driving force of the evolution.
Okay. Yeah, and the fact that we have a lot of viral DNA in us that that has been endogenized. We're part virus. It's weird.
Yeah. Yes.
They've guided our evolution quite a bit.
Mm-hmm.
So okay, well, very good. Lili, what's the best way for people to find out more about your research and your instruction? Where can they go?
Just go to our lab's website and you can find me. And if you are interested in me, you can just go to PubMed or Google. Just type in Lili Wang, HIV cell to cell, and you'll find papers there.
Excellent. Lili, thank you for coming on the podcast. I really appreciate it.
No problem. I'm happy to share my thoughts, although some of them might be not so mature.
It's okay. It's okay, yeah.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.